Hello, everyone, and welcome to this lecture on comparison of nuclear DNA and Y chromosome DNA in forensic application. In the field of forensic science, DNA has become one of the most powerful and reliable tools for human identification. From solving complex criminal cases to identifying victims in mass disasters, DNA profiling plays a vital role in uncovering the truth. But did you know that not all DNA used in forensic investigations is the same? Two important types of DNA, nuclear DNA and Y chromosome DNA, each serve distinct purposes based on the case scenario, the type of biological sample, and even the gender of the individuals involved. Nuclear DNA is present in nearly every cell of the human body and is inherited from both the parents. Its high variability depends between individuals makes it extremely valuable for personal identification. On the other hand, Y chromosome DNA is found only in males and is passed down almost unchanged from father to son. While it does not have the same individualizing power as nuclear DNA, it's incredibly useful in specific cases, especially when only male DNA needs to be analyzed, like in sexual assault investigations involving mixed samples. So, in this lecture, we will explore how these two types of DNA differ in structure, inheritance, and forensic utility. We will also look at the strength and limitation of each and understand how forensic experts decide which type of DNA to analyze depending on the case. Let's now talk about nuclear DNA, the most commonly analyzed form of DNA in forensic science. Nuclear DNA, as the name suggests, is located in the nucleus of nearly every cell in the human body, whether it's from blood, skin, hair root, saliva, or bodily fluid. It is the complete genetic blueprint of an individual. Nuclear DNA is inherited from both parents, 50% from the mother and 50% from the father. This biparental inheritance results in a unique DNA profile for every individual except in the case of identical twins. Structurally, it consists of 22 pairs of autosomes and one pair of sex chromosomes, XX in females and XY in males, making up a total of 46 chromosomes. One of the most important characteristics of nuclear DNA is its high degree of variation among individuals. This is due to genetic recombination, a natural process during reproduction that shuffles genetic material from the parents, creating a unique genetic identity in their offspring. This uniqueness is what makes nuclear DNA an extremely powerful tool for forensic identification. It allows forensic scientists to match DNA samples from a crime scene with suspects or identify unknown victims with remarkable accuracy. In addition to forensic casework, nuclear DNA is also widely used in paternity testing, missing person investigations, and mass disaster identification. So, when forensic experts are looking for the most definitive match, nuclear DNA is usually their first choice. Now let's understand Y chromosome DNA, a specialized form of DNA with unique forensic significance. The Y chromosome is one of the two human sex chromosomes and is found only in males who carry the XY chromosome pair. Females, on the other hand, have two X chromosomes and therefore do not possess Y DNA. What makes Y DNA particularly interesting in forensic genetics is its pattern of inheritance. The Y chromosome is passed almost unchanged from father to son, generation after generation. This means that all male members of the same paternal lineage, such as brothers, uncles, fathers, and grandfathers, share very similar Y chromosome DNA profile. Unlike nuclear DNA, which undergoes genetic recombination during reproduction, the Y chromosome does not recombine except in very small regions called pseudo-autosomal regions. This lack of recombination helps preserve the Y chromosome structure over generations, making it a powerful tool for tracing paternity ancestry and linking individuals from the same male line. In forensic casework, Y chromosome DNA becomes especially valuable in male specific investigations, for example, in sexual assault cases where female and male DNA may be mixed. YST analysis allows forensic scientists to isolate and analyze only the male component, even in the presence of overwhelming female DNA. However, it is important to note that Y DNA cannot distinguish between male relatives on the paternal side because they share nearly identical Y profiles. This limits its individualization power, but not its useful as in narrowing down suspect or confirming paternal relationship. In summary, Y chromosome DNA offers a unique lens into the male lineage and plays a crucial role in forensic analysis where male identification is key. So let's now compare nuclear DNA and Y chromosome DNA side by side in terms of their forensic treatment. Nuclear DNA is inherited from both parents, while Y DNA is inherited only through the paternal lineage. Nuclear DNA is found in all nucleated cells and is present in both males and females. In contrast, Y DNA is found only in males and only in their nucleated cells. In terms of variation, nuclear DNA provides a unique profile for each individual, making it highly discriminatory. White DNA, however, shows low variation as all paternal male relatives typically share the same profile. Nuclear DNA is used for general forensic identification, while white DNA is applied in male specific cases, especially when isolating male DNA is necessary. In mixed DNA samples, nuclear DNA analysis can be complex, especially if profiles overlap. White DNA, however, is especially useful in male female mixtures, helping to isolate the male component. Finally, nuclear DNA uses markers like STRs and SNPs, while white DNA analysis primarily relies on YSTR markers.
Applications of nuclear DNA in forensics. Now let's explore the key applications of nuclear DNA in forensic science. One of the most important uses is personal identification. Because nuclear DNA is highly individual specific thanks to genetic recombination, scientists can create a unique DNA profile for nearly every person. This makes it a powerful tool in criminal investigations, helping match suspects to evidence left at crime scenes. Nuclear DNA is also widely used in paternity and kinship analysis. Since a child inherits half of their nuclear DNA from each parent, the information can be used to determine biological relationships. It plays a critical role in legal disputes over child custody, inheritance, and immigration cases requiring proof of familial connection. In criminal casework, nuclear DNA is essential for solving serious crimes like rape, murders, assault, and burglary. It can help confirm the presence of a suspect at a crime scene or exclude someone who has been wrongly accused. Even a small trace of biological material like blood, saliva, or hair root can yield a match.
Another major application is in missing person investigations. Nuclear DNA from relatives of a missing person can be compared with unidentified remains to establish identity even after years have passed. Finally, in tragic situations involving mass disasters such as earthquakes, fires, plane crashes, or terrorist attacks, nuclear DNA is used to identify victims when physical recognition is impossible. It brings closer to families and assists with legal documentation such as death certificates. In all of these scenarios, nuclear DNA stands out for its high accuracy, reliability, and power of discrimination, making it the cornerstone of forensic genetics.
Now, let's take a look at the application of Y chromosome DNA in forensic science, particularly in situations where identifying or isolating male DNA is essential. One of the most common applications in sexual assault cases, especially those involving a female victim and a male perpetrator. In such cases, biological samples often contain a mixture of male and female DNA. Traditional nuclear DNA profiling can be complicated when female DNA is in excess. Y chromosome analysis allows forensic scientists to target and analyze only the male DNA, even when it's present in very small quantities. White DNA is also extremely useful in mixed DNA samples, such as those recovered from clothing, swabs, or touch evidence. When both male and female cells are present, YSTR analysis can effectively separate and highlight the male genetic material, helping to narrow down the pool of potential suspects. Another key application of Y chromosome DNA is in tracing paternal lineages. Because the Y chromosome is passed down almost unchanged from father to son, it can be used to explore historical connections, surname studies, or to assist in identifying missing persons by comparing DNA from paternal male relatives. Additionally, Y-DNA analysis can help in the exclusion of suspects in cases where the DNA mixture is male specific. For example, in gang related assaults or crimes involving only males, if a suspect's Y-STR profile does not match the profile from the crime scene, they can be confidently excluded. In summary, while Y chromosome DNA cannot provide individual identification. Like nuclear DNA, its ability to isolate and trace male DNA makes it a valuable tool in specific forensic contexts, particularly in cases where other forms of DNA analysis fall short. Case example sexual assault with mixed DNA. Let's consider a real world case scenario where Y chromosome DNA made a crucial difference. In a sexual assault investigation, forensic experts recovered DNA evidence from a female victim. The samples contained a mixture of both male and female DNA, which made traditional nuclear DNA analysis difficult. To overcome this, scientists used YSTR profiling, which specifically targets DNA found only on the Y chromosomes. This allowed them to isolate the male component from the mixture despite the overwhelming presence of female DNA. The resulting YSTR profile was then compared against suspect samples. It either excluded innocent individuals or helped positively identify the perpetrator, narrowing down the investigation and supporting legal proceedings. This case highlights how Y chromosome DNA can be a powerful tool in male specific forensic cases, particularly when standard DNA analysis doesn't provide clear answers. Thank you for watching this lecture on the comparison of nuclear DNA and Y chromosome DNA in forensic applications. We hope this gives you a clear understanding of how both types of DNA are used in solving crimes, identifying individuals, and serving justice through science. If you found this video informative, please like, share, and subscribe to this channel for more content on forensic science and genetics. Thank you.